Hi everybody, welcome to Chapter 3, Microbiology Basics for Endoscope Reprocessors. Lucky for you, you've already taken microbiology, so most of this should be a review for you. However, we will talk about specific types of infections that occur due to a specific type of endoscope procedures. So that is at the end of this lecture, which is really important, the different types of infections that can occur because of pathogens, viruses, bacteria that have left on these endoscopes unintentionally between being processed. So again, this chapter should be a little bit of a review for you, but it's also a good review and preparation for your certification exam. So pay attention, take some notes, and you will have an assignment that corresponds with this lecture. So upon exiting this chapter, we will have discussed the importance of protecting patients from infection, review basic concepts of microbiology, discuss biofilm, identify infections associated with specific endoscope procedures, as well as survival of pathogens on environmental surfaces and cloth. All right, a brief introduction. So the main purpose of reprocessing in general is to remove microbes and bioburden following patient use. For endoscopes, again, it is extremely important because the risk of infection is so high. So we need to be meticulous when we are decontaminating endoscopes, and as well as wearing proper PPE. Now that's not just for patients, that is also for us. It's also crucial to use the most appropriate products, best practice, and most effective techniques. What this means is that one type of high-level disinfectant might not be appropriate to kill all of the germs we need it to for a specific type of endoscope. So it's important that we know what to use and why we're using it. So on this slide, we've got a couple of key terms that are going to be important for you to know. Again, hopefully a little bit of a review, but I would like for you to take a few minutes and write down the definitions to these terms: microorganism, bacteria, symbiotic bacteria, infection, and pathogens. So the more aggressive a pathogen, the fewer of them are needed to cause an infection. The physical condition of an infected person can play a role in how severe the infection can become. So we talked about that a little bit when we talked about chain of infections and our most vulnerable patients. So this is not only this isn't excluding anybody, but we do know that there are more vulnerable patients that are more susceptible to contracting some of these viruses. Those are infants, injured people, elderly, and anyone with a compromised immune system. So the elderly and the infants have a very low immune system. One hasn't really kicked in yet, and the other one's just slowly declining as you get older. If you are injured, you are already fighting to a certain degree wherever that infection or your injury is. So your body's already in high motion trying to heal it. So it doesn't have a whole lot of reserve for another type of infection. And that is the same with anybody who has a compromised immune system due to other pre-existing factors, cancer, HIV, AIDS, any of those immune compromised types of patients have a higher potential of contracting some sort of infection. Couple of pathogens on this page. So fungi, we've got a diverse group of microorganisms and plants. We also have viruses, and that's a group of infectious agents that can only reproduce by using mechanisms inside of a living cell. And then we have parasites that need a host in order to survive. So here are some other key terms that you should be very familiar with by now, and if not, please take some notes and write those key terms and definitions down. All right, so how do we detect bacteria? Well, we do it through a couple of different ways. So we have different types of staining. The most popular is a gram staining. If we have a gram-positive type of bacteria, it is going to show a purple stain, and gram-negative is a red or a pink. We also have acid-fast staining. Make sure that you are familiar with the different types of staining and the different colors. Those have been known certification questions. Well, might not be a lot of them, but there are definitely a few. As far as gram-positive is purple, gram-negative is pink or red. Endospore stain, which turns those endospores and spores green. This is one of the ones that is especially difficult for us to kill, and it's the one that our biological tests test for to make sure they have been destroyed after the sterilization process. They do not need food or water to survive, so they are actually extremely difficult to kill because they don't need a whole lot. All right, some different types of gram stain bacteria. So gram-positive bacteria, you've got your strep, your staph, Clostridia, which lives in your colon. And then you have your gram-negative bacteria, your E. coli's, your Helicobacter pylori, your Neisseria. Make sure that you are aware of what type of bacteria falls under positive or negative. So fungi, back to talking about fungi. So branches of fungi can provide a shelter, so they can work in conjunction and help that bacteria or virus thrive by providing a shelter for it to hide underneath. There are different types of fungi, but Candida can cause thrush, which is a very uncomfortable type of rash that either babies get or even adults. It's typically found on the tongue. It's very uncomfortable. Fungi is also responsible for diaper rash in babies. All right, we've got our viruses. They are much smaller than bacteria. They need a living cell to be able to reproduce. Between our viruses, we have an enveloped and a non-enveloped virus. Our non-enveloped viruses are actually the most difficult to kill. So that's an important fact for you to know, that we have different types of viruses, enveloped and non-enveloped viruses. Biofilm, we've heard this term before. It's the same as it is over in our other types of books. It is all the same. Biofilm loves to start in little scratches and gouges in our instruments because it's a great place for them to hide. It decides to call up its friends, they hang out, they release this protective gel over that area, and then one friend from this gel colony moves on to the next area and it forms its own colony there and starts inviting everybody to join in. So it moves very, very quickly and it leaves a protective gel over our instruments, which we cannot see, but it's very difficult for our enzymatics and our detergents to break down. So the presence of biofilm is always possible. So that's why we need the reprocessing of the instruments to start as soon as possible after use. All right, so the next few slides are going to discuss different types of infections that are related to endoscope procedures. They are very specific to the area or the anatomy where the procedure is being performed. So the first one is your bronchoscopy, that is looking at your lungs, your bronchioles. So by nature, you might have some inflammation of that area. So for midterm itis is inflammation in bronchos or the bronchioles from your lungs. So that is a potential infection, as well as pneumonia, as well as sepsis. For colonoscopies, you have the potential to have an infection in your appendix, the peritoneum, which is the area surrounding your intestines, as well as endocarditis, which is a heart valve infection. So that occurs because that is a very vasculated area. If it's picked up in the bloodstream, it will carry it all the way to the heart. So your cystoscopy and your ureteroscopy, potential for infections are a urinary bladder infection because that is exactly where this procedure is being performed. kidney infection, as well as endocarditis. For the procedure of a duodenoscopy, you have the potential for gastroenteritis. Itis, again, inflammation. Gastro is stomach, enter is intestines. So inflammation within your stomach or intestines. Sepsis, which is full body shock, is also possible, as well as hepatitis C. For upper gastrointestinal endoscopies, so talking about your stomach specifically, the potential for different types of infections are bacterial peritonitis, endocarditis, gastritis, and sepsis. So why do we have the potential for these infections? Well, a few different types of reasons. The one is lapse in endoscope cleaning and disinfection. So if you miss a step or you're not performing best practice for these type of medical devices, the potential for infection is extremely high. If you are not correctly or insufficiently cleaning and disinfecting per those manufacturers' IFUs, that is also a reason for an infection. We simply have some poor endoscope designs. Endoscopes are not perfect, and for them to do their job, they have to be designed in a way where they're functional, but it also makes it very, very difficult for us to clean. So sometimes these things are a little unavoidable in the sense of we can't reach all the areas based on the anatomy of the instrument. If we have a contaminated AER or accessories, that is going to create the potential for infection, as well as we use contaminated rinse water or any type of contaminated surfaces. Last but not least, on this slide, we have the difficulty of killing pathogens from the easiest to the most difficult. So on this list right here, it starts with enveloped viruses. That is the easiest for us to kill. All the way down at the bottom, you'll see bacterial spores. Well, we know those are the most difficult to clean because that's what we test for in our sterilizers. So we know that if we test for the most difficult to kill, and the sterilization process shows that we have indeed killed those bacterial spores, then everything on this list before that has also been killed. In conclusion, flexible endoscopes and reprocessing of these types of endoscopes is extremely difficult. They are not your typical surgical instruments that you're used to seeing. They require a tremendous amount of work, focus, knowledge, and attention to detail. As long as you are following best practice methods, then you are on your way to delivering an endoscope to the patient that is safe for use.